Good morning and welcome to ENDO 2015. My name is Tom Zeller. I'm from the University of Massachusetts at Amherst, and I'm going to be moderating this press conference. This conference is really focused on endocrine-disrupting chemicals, or EDCs, and we've got 4 talks that are going to be highlighted from all the different presentations that are going to be given during this 2015. The Endocrine Society has had a long-standing voice in the science of EDCs, in part because in 2009 we published the first scientific statement that really made a big difference really around the world. And it's reasonable that we would have this kind of global effect because members of the Endocrine Society come from 100 different countries. 40% of the membership comes from outside the United States. So we have a very global scientific voice, and it's reasonable that we have that. Endocrine disruptors, EDCs, are becoming important not just because they're in vogue, but also because what members of the Endocrine Society and other scientific societies are showing is that these chemicals can interfere with hormone action in a way that produce adverse effects at levels that don't produce toxic effects. So we really need to discriminate between overt toxicity and effects on endocrine systems that produce abnormal and adverse outcome. So we're going to have 4 talks today, and the way this is going to run is the first talk is from Leonardo Trasande, who's a pediatrician at the New York University School of Medicine. He spearheaded a large group of people in really an amazing way to produce a series of papers to estimate the burden of disease and cost of EDC exposures in Europe. And this is really the first of a kind, a very important set of observations and analytics. At the end of his talk, we'll have a special question and answer, and I'll kind of moderate that as we go. There are 3 other talks after that first question and answer period. These talks were selected over in association with the way these abstracts are being reviewed, and there are 3 additional important issues that are being highlighted here. The first is Dr. Andrea Gore, who's the Johnson & Johnson Centennial Professor at the University of Texas at Austin in the School of Pharmacy. And what she's going to talk about is the transgenerational effects of EDC exposure. This is becoming a very important issue and has important implications for public health. Then we'll hear from Dr. Jennifer Adibi, who's professor at the University of Pittsburgh, and she's going to talk about a really interesting study where she's linking EDC exposures to placental function and adverse outcome in male reproductive development. Finally, we'll hear from a doctoral candidate, Stephanie DeGroot, from Quebec. And she's actually produced a study that demonstrates that 2 chemicals, 2 endocrine disruptors that have no overt effect on autistic-like behaviors, when given together, produce autistic-like behaviors in an animal model of autism. These are extremely important implications, and this is why they're going to be highlighted here. So without further ado, I'll turn it over to Dr. Trasande.
Thank you, Tom. It's a privilege to be here today to present work by 18 scientists from 8 countries across the world who worked intensively over 15 months to produce this work. I'll begin by summarizing the highlights of our main findings. We identify 13 chronic conditions with strong scientific evidence for causation by endocrine-disrupting chemicals. Based on current knowledge, the portion of those diseases that can be directly traced to endocrine-disrupting chemicals cost Europe 157 billion euro. That cost could actually be much higher, on the order of 270 billion euro. What's important to highlight about this work is that it represents a small subset of the chemicals that are actually EDCs and may contribute to human health and disease. While future work will consider other health consequences, especially those in women, we examined a subset of the diseases that can be linked to endocrine-disrupting chemicals. And also, we identified only a subset, we quantified only a subset, that is, of the economic costs that can be traced back to these EDCs. Despite all of those limitations, which suggest that this is a subset of a larger economic cost, potentially in the trillions of dollars, limiting our exposure to the most widely used and potentially hazardous EDCs represents a substantial public health and economic benefit. First, some context. Here I present the European Union definition of endocrine-disrupting chemicals, which was the focus of our report, though we also considered very strongly the Endocrine Society's own definition of EDCs. Suffice it to say, while there's variability in the definition, the highlights are that endocrine-disrupting chemicals disrupt hormones and produce disease. They are a highly heterogeneous group of molecules as listed here, with a substantial, diverse, and ubiquitous use. The first observation that EDCs contribute to human health and disease was the observation by Herbst and Byrne in young girls exposed in utero to the pharmaceutical diethylstilbestrol, who suffered cancer many decades later as a result of that early life programming impact of that chemical exposure. Subsequently, rapidly accumulating evidence suggests that EDCs contribute to a host of conditions that span across the life course. For EDCs, across the host of conditions that have been linked etiologically, there's varying evidence, epidemiologically, that is in populations, and toxicologically or in the laboratory setting, with the complicating factor that each of these conditions is ultimately the byproduct of many diverse factors, including diet, physical activity, genetics, just to name a few. Yet, in the absence of estimates of the health and economic consequences that can be attributed directly to EDCs, the high costs of safer alternatives are likely to outweigh any concerns about ongoing use of EDCs. The European context here is very important. In 2009 and 2011, landmark legislation mandated limits on pesticides and other chemicals with endocrine-disrupting properties, geared directly with a focus towards those that had health and environmental effects. Currently, the European Union is requested an impact assessment which considers the economic consequences of EDCs as well as the costs of safer alternatives. And so our objective was to present a range of health and economic costs that can be reasonably attributed to EDCs in the European Union. We convened first a steering committee that identified 3 panels focusing on the conditions for which the evidence was strongest for causation, specifically obesity and diabetes, male reproductive consequences, as well as neurodevelopmental deficits and disabilities. 2 expert panels were also convened for breast cancer and female reproductive conditions and will be the basis for future reports. The panelists were selected based upon their scholarly contribution. We used a highly cited and widely used approach, first described by the Institute of Medicine in 1981, to identify the attributable portion of diseases and associated costs that could be traced back to EDCs. We applied data from human studies along with the most representative European biomarker data available, and we selected only peer-reviewed published cost data for these diseases, which represent, therefore, a conservative estimate because they do not consider potentially, in many cases, lost work and other social suffering associated with these conditions which have a value to society. In estimating the attributable portion of EDC costs, we were cognizant of the reality that there is uncertainty. And we, in the context of that uncertainty, leveraged the landmark work of the Intergovernmental Panel on Climate Change. Panels were instructed to evaluate the epidemiologic, that is the population or human evidence, together with the laboratory evidence, but they were instructed to do so using rigorous criteria separately. Together, that population evidence as well as the laboratory evidence was assembled to identify a probability of causation, a percentage probability of causation. And these percentages were used to downweight the cost and diseases that were linked to EDCs. Across the 13 EDC health outcome relationships that we identified, there are a range of probabilities. And there are extreme scenarios that you can imagine. That is, if you proverbially flip a coin 13 times, there is a substantial but very modest probability that you will get 13 heads or 13 tails. But in practice, that's not a realistic range of possible scenarios. And so we used very widely used Monte Carlo modeling to produce a realistic range of probable costs across all 13 EDC health outcome relationships. The subsequent slides here represent our results for individual chemical categories. For pesticides, we identified the greatest costs, with 13 million lost IQ points linked to early life organophosphate exposure with associated substantial intellectual disability. We also identified childhood obesity with substantial, though less, economic costs linked to that condition, as well as adult diabetes. For phthalates, chemicals used in food wraps, as well as cosmetics, shampoos, and vinyl flooring, we found a variety of consequences, most especially decreases in testosterone with associated early cardiovascular mortality, with substantial, roughly €8 billion loss economic productivity. In addition, we identify substantial additional need for assisted reproductive technology due to male factor infertility, with additional costs in the range of $4.7 billion, and also identified substantial adult obesity as well as adult diabetes that could be etiologically traced to phthalate exposures, with costs in the range of €16 billion. For brominated flame retardants, we found much more modest but substantial impacts on the developing brain, with roughly 8 billion in lost €8 billion, that is, in lost earning potential, as well as an additional €2 billion in intellectual disability associated costs. We also identified modest contributions to testicular cancer as well as undescended testis, with associated costs in the range of €1 billion. Finally, we also identified substantial contribution to neurodevelopmental disabilities, including autism and attention deficit hyperactivity disorder, with costs in the range of €1.9 billion. In addition, and finally, we found associations of bisphenol A with childhood obesity, with subsequent costs in the range of $1.5 billion. For each of these conditions, the percentage contribution is quite modest, but the economic impacts are quite substantial. Across all these 13 conditions, we found a range of potential economic costs in using the Monte Carlo modeling. Across 1,000 modeling scenarios, which considered the variability and probability of causation, the average cost was €157 billion. This slide represents a distribution of those costs by chemical as well as by disease category. At the extreme, the cost could be as high as €270 billion, assuming all of the associations are actually causal, though in practice, the probability that this is the case is quite modest. That said, there's also a greater than 99% probability of that at least one EDC health outcome relationship is real, suggesting that it is extremely likely that EDCs contribute to disease and disability in Europe. The implications for the United States are substantial. Given that I'm speaking here in San Diego, I'll take a minute and reflect on that. A similarly large burden of disease may also be attributable to EDCs in the United States insofar as exposures are similar. And indeed, data from the Centers for Disease Control and Prevention confirm that such exposures are in the similar range. And so the costs of EDC exposures in the United States are likely to be in the same order of magnitude. That said, that is the basis for future research. There are differences, though, in regulation between the United States and Europe. And insofar as brominated flame retardants are more closely restricted in their use in the European Union, the costs of brominated flame retardants in the United States is likely to be more substantial. Indeed, in the United States, there is a good story to describe insofar as levels of certain phthalates, including di-2-ethyl hexyl phthalate, the most widely used phthalate in food wraps, for example, have decreased substantially over the past 10 years. And the cost of attributable disease in the United States may have also decreased substantially over that same time span. More generally, though, EDCs are used across the globe, including in developing countries where regulatory frameworks are more modest, and our findings also support careful regulation as part of the strategic approach to international chemicals management. And so just to review once again, we've identified 13 chronic conditions with strong scientific evidence for causation, with costs in the range of 157 billion euro, which corresponds to 1.23% of GDP. What's important to emphasize once again is the narrow subportion of the actual costs that this estimate represents. We examined less than 5% of EDCs. We examined only a subset of conditions linked to those EDCs, and we only examined a subset of economic costs. And so limiting exposure to the most widely used and potentially hazardous EDCs is likely to produce substantial economic benefit. I'd like to thank the funders who supported this work, as well as my colleagues across the globe who contributed to this landmark effort. Thank you very much.
Thank you. We can take questions both from the audience here, but also in Brussels and online. Make sure that you use the microphone if you're going to ask a question here.
And please state your name before you ask your question.
Ron Cheng. A question on something. You didn't get into actually the epigenetic modulation that's happening in the parts of the brain in terms of how neurons are possibly not activating their axons, and how various axonal connections are being dysregulated, and how these super circuits then actually manifest in terms of behavioral effects that are just regulatory to the organism.
I agree that we only did that indirectly, though epigenetic effects of these EDCs were identified as mechanistic inputs that supported the strength of evidence evaluation, both in the laboratory and in humans. So it was supportive evidence, though we didn't. We clearly need further research to identify the mechanisms by which EDCs contribute to disease and disability. Yes.
And I have a few questions from the webcast. Do you have any estimations about the cost of EDCs for the health service in the U.S. and or Europe?
Can you repeat the question just so I understand it better?
Do you have any estimations about the cost of EDCs for the health service in the U.S. and or Europe? It's a bit confusing of a question.
I'm a bit confused by the question insofar as the health service. I believe the presentation reflects on the comparative effects of EDCs in the United States and Europe with potentially disparate economic consequences. Again, these speak to the importance of policymaking insofar as it can vastly influence EDC exposures.
Okay. And this one, maybe, Tom, you might want to weigh in on as well. How would you judge on the EPA endocrine disruptor screening program? Is it, in your view, an appropriate way to address the issue?
So, first of all, the EPA has a hard job because there are about 84,000 chemicals in commerce, and the way it has been traditionally approached, it would just take a lifetime to be able to go through all those chemicals and ensure safety. So they do have a hard way to go. On the other hand, the kind of validated screens and tests of the endocrine disrupting screening program, the way it's designed at this moment, is really a 50 to 80-year-old set of assays that I think are really not comprehensive. and they don't really take into consideration principles of endocrinology that are really required to ensure that these chemicals are being evaluated appropriately. I think the EPA understands that, and they're using what they're calling ToxCast or computational toxicology to address it, but it's going to take a while to get there. I think at this moment, one of the most important sources of information that we can use to protect public health is academic science that's coming from members of the Endocrine Society as well as other professional societies.
And then a question for Dr. Trisande. Just to clarify, the cost estimates that you show in the beginning, are you saying that these are conservative estimates?
There are three major reasons why these estimates are extremely conservative. We examined fewer than 5% of EDCs. We examined only a subset of health outcomes, and we only examined a subset of the economic costs traced to the diseases that we actually measured costs.
Basically, you're pointing out one of the most ubiquitous issues facing human evolution. What can we do about the fact that everything we do, from what we eat, from the air we breathe, from everything that surrounds us, is the use of extraneous chemicals that were not concordant with the evolution of the hominoid species, which we are. So what do you do? I mean, what is the fact that the only way to abort this would be to move to some very rural area outside of the United States and grow your own food and live in a way that Thoreau talked about in Walden Pond, a way to abort this?
We believe this data presents an opportunity for more balanced debate on the ongoing use of endocrine-disrupting chemicals and trade-offs with safer alternatives. And so far as this is the first effort to quantify the costs of EDC exposures presently, we can finally have that as a counterfoil to the costs of safer alternatives.
Lynn Peterson with Trends in Medicine. So are there alternatives, or is this a cost we have for something we're actually, for fire deaths, if we go back to before flame retardants? Do we lose more children and people than we are losing from this? Did you look at that?
There are in many cases safer alternatives to these chemicals, but more fundamentally, we have a regulatory framework, at least in the United States, that doesn't rigorously screen chemicals for their potential effects on the endocrine system and on human health. And if we do not have that regulatory structure in place, the risk we can run is that the replacement may be as problematic, if not more, than the current EDC use. That having been said, these data speak to the need for further action to eliminate the health effects associated with EDCs in Europe and across the world.
If I could add to that, too, it's interesting that you bring up flame retardants because they haven't been shown to protect property or lives. So, you know, they've been used for quite a while. These data are out there, which is why Europe doesn't have the same stringent kind of code of use of flame retardants that we have in the United States. So I think that thinking about the necessity, the requirement, what are these chemicals intended to do? Do they actually serve the purpose on a population level? These are important questions that really need to be addressed kind of honestly and openly.
All right, and just a few more questions because we'll want to make sure we have time for the other presenters. So if you do have questions after these few, please feel free to hang around here and ask Dr. Trisande your questions. So here's another one. Is there any thought to applying this methodology to estimate costs in the United States?
As I indicated, clearly a natural next step is to extend this work to the United States context.
And how do you explain that the FDA came out recently to say that BPA use in cans is safe?
Our work accounted systematically for the uncertainties that exist in the evidence, and so far as there were modest concerns about the effects of BPA on obesity in children, we downweighted the costs and disease burden by that probability. And so clearly there's a need for greater research in this area to better disaggregate the effects of EDCs on human health. But our work speaks to the substantial evidence for BPA effects on human health.
All right, and then we have one last one, and this is what sources of information did you use for the health costs, given that there are very different situations, health problems in different EU countries?
That's a great question. Thank you for asking it. Whenever possible, we tried to obtain data from individual EU countries, and there are 27 of them with diverse and disparate intensities of health system delivery. Insofar as that was feasible, we did just that, but we acknowledge that for many countries, there were no cost data available. And so, for example, we would apply German data across the entire EU. Insofar as we were limited in our capacity to get country-level data, there is a modest amount of uncertainty in those analyses. But in general, given the substantial healthcare infrastructure across the entire EU 27, our economic costs estimates are quite solid.
Okay, so the next speaker is Dr. Andrea Gore, who's also the editor-in-chief of the journal Endocrinology. I'll let you cue that.
Do it. I'm not a PC person. Let's see. Here it is. Okay. So thank you for the invitation to speak, and I'm going to ask everybody here now to put on your basic science hat, because I'm going to be speaking about some experimental data that we've been conducting in the lab on how exposures to low doses of environmental endocrine-disrupting chemicals, or EDCs as I'll be calling them, during pregnancy affect not only the exposed fetuses, but also 2 subsequent generations. Just a little bit of background about EDCs in the developing brain. The brain is an endocrine organ, and it's one of the most sensitive parts of the body in terms of both natural hormone sensitivity as well as EDC sensitivity. The group of EDCs that we study in the lab are called polychlorinated biphenyls, or PCBs, which were used in industrial applications for many years, and although have been banned around many parts of the world, they're quite persistent. They're found in virtually every country, all water systems. They're in the air, they're in the soil, and they're in almost every person's body to this day. PCBs have been known for a very long time to be environmental EDCs, and they act upon many of the same pathways that natural hormones such as estrogens, androgens, thyroid hormones, and others act. I mentioned that the brain is highly sensitive. One of the reasons is because it expresses receptors for all of these different kinds of hormones, and also neurotransmitters, processes involved in neurodevelopment, are also very sensitive to hormones and to hormone disruption. So we're most concerned about exposures to EDCs during early periods of development, especially in the fetus and in the infant, when the brain undergoes a tremendous amount of growth, synapses form, connections are made, cells are born and die. And so our work is focusing on a hypothesis called the developmental origins of health and disease, or DOHAD, where we look at low-dose exposures to endocrine disruptors prenatally and determine the outcomes on the developing brain into adulthood. And in the study, we're also going to talk about these transgenerational effects to second and third generational animals that are born to the originally exposed animals. So we have a bit of a complicated model, and I'm going to talk you through it quickly. What we do in the lab is we work with rats. So here we have a pregnant rat, and we expose her to low levels at the beginning of the 3rd trimester of pregnancy, either with a PCB mixture called Arichlor 1221 or A1221. We also compare the effects of A1221 to estradiol itself, since we know some PCBs act through estrogen systems. And we also have, of course, a vehicle control group. We allowed those animals to give birth, and they have male and female pups. The animals look perfectly normal. There's just this very small exposure. And then we breed these animals. So we call this, sorry about the cursor, we call this the F1 generation. We breed through the females. We breed a maternal line down 3 generations to the F2 and the F3 generation. We also breed the males to the F2 and F3 generation. And this is an important point because the effects of the EDCs that we see sometimes differ depending upon who your parent or who your grandparent was. We look at development, hormones, and I'm going to talk a little bit about some of the data in the brains from these animals. I'm now going to show you some very complicated slides, but I'm going to talk you through it. They're going to be organized similarly to here, where we'll show the F1 generation, then the F2, and then the F3 generation through maternal and paternal lines. So I'm going to talk you through the first one slowly, and then I can go through the others pretty quickly. So my first point that I'd like to make is that consistently the body weight is increased in the lineages of animals that were exposed to EDCs, and it's transmitted primarily through the maternal line. So if your mother was exposed, and actually I'm sorry, this pointer just isn't working. Here it is. The F1 generation shown here, these are the females, their body weight. In blue is the control group. In green is the estrogen group, and in red is the PCB Erichlor 1221 group. These groups are significantly heavier than these animals, although they were raised absolutely all the same after that prenatal exposure. We don't see any effects in the male F1 generation. We then look at the offspring, the F2 and the F3 generations, across the maternal and paternal lineages. What you can see in the females through the maternal lineage is that this PCB group, shown in red here with the star, is still significantly heavier than her counterparts. There's a bit of an increase in the F3 generation, but it's not significant. No effect if you were bred through the paternal line. In the males, we see both in the F2 and the F3 generation heavier animals that derived from the original EDC exposures, but not through the paternal line. And these data are consistent with human data and with other EDCs showing that body weight, obesity, diabetes, and diseases associated with EDC exposures happen not only in exposed animals, but in their descendants. I'd also like to show you one slide on hormones. We measured several hormones in these animals. I'm just going to show you levels of estradiol, which is the major estrogen in both males and females. I'm going to show you the female data. And what we found were that the PCBs increased estradiol levels, but we didn't see that till the 3rd generation. So here's the F1 generation again, no difference between the 3 groups. F2 generation, no differences. F3 generation, significantly higher estradiol levels, irrespective of which lineage you came from. Finally, I'll show you 2 slides on our brain data. I just took a subset. We actually looked at 96 genes in the brain, part of the brain called the hypothalamus arcuate nucleus, which is very important for metabolism, energy balance, circadian rhythms. Here we looked at a gene called CRH1, or corticotropin-releasing hormone, which is involved both in stress and in energy balance. And what we found was that CRH1 levels were increased in the F3 females and in the F2 males, again, just through the maternal generation. So here are the males. Actually, it was just for estrogen in the males, in the F2 males. For the females over here on the bottom left, you can see that gene expression is higher in the estradiol and PCB groups. Finally, I'll show you one more set of genes. These are genes that are very important for circadian rhythms. I'm just showing you the data for the males for 2 different genes. One is, they're both circadian genes. One's called period 1, and one is called clock. And these happen to be affected just by estrogen, but you also see it just in that F3 generation. So let me try to give you a few take-home messages about this data set. Exposures to low levels of PCBs that are comparable or even in some cases lower than what humans are exposed to affect body weight, hormones, and expression of genes in the brain. Although I haven't shown you any of the data, in other studies that we've published on other EDCs, we've also shown behavioral changes in F1, F2, F3 animals. We've shown that there's epigenetic mechanisms of transmission of these traits across the generations. Second, the body weight and the hormone effects are actually more pronounced 2 to 3 generations later. So think about from the cost of from thinking of the analysis of the costs of EDCs, we're looking at exposed people, but think about the further costs to their children and their grandchildren that cannot even be estimated. We also found that the gene expression changes in the brain were most pronounced in the first generation, which is to be expected. They were the exposed animals, but many persisted all the way to the F3 generation. And these genes included biological rhythm genes and body weight regulation genes and circadian genes. And who your parent was and who your grandparent was matters. Was it through the grandmother? Was it through the grandfather? Your sex also matters on the outcome. So I'll stop there. I know we won't have questions until the end, but I'd be happy to take them at that time. Thank you.
So the next speaker is Dr. Jennifer Adibi from the University of Pittsburgh.
Yeah, thank you very much, Tom. So I'm going to talk about a pregnancy hormone that's produced by the placenta called human chorionic gonadotropin, and an analysis that we did to understand its relationship to sex-specific development of the fetus and the response to an endocrine disruptor. So we studied the exposures to a class of chemicals called phthalates. Phthalates are found, as Leo mentioned, in plastics, personal care products, food packaging, furniture, to name a few. In U.S. studies, they've been measured in 95 to 100% of urine samples, and this is true for pregnant women as well. And the growing body of human studies are showing us that prenatal urinary concentrations in the mom are associated with sex-specific outcomes in the child. Two types of outcomes that have been characterized and shown to be associated are, one, the distance from the anus to the genitalia in the male newborns, and secondly, measures of intelligence and behavior going out to 7-9 years postnatal life. So this pregnancy hormone, it's been established, and we also studied this in the California population, that women carrying female fetuses have about 10% higher levels of this hormone than those carrying male fetuses. And we actually don't really know why this is or what the meaning of this, the significance of this is in terms of the biology. And a key thing to know about this hormone is it has been established that it's essential for this hormone to bind to a receptor in the fetal testis and the male testis to stimulate testosterone production and therefore normal male development. And so the placenta, as I mentioned, is the source of this hormone. And this is a beautiful image of the placenta in the first trimester of pregnancy. And as you can see, it's this large, bushy mass of tissue that surrounds the early embryo and the gestational sac. And our general hypothesis is that the phthalates in the maternal blood are hitting the placental tissue and then changing expression of key hormones, proteins, other types of molecules expressed by the placenta that then reflect back into maternal blood, and we can measure this at the population level. And likewise, this essential hormone, the levels could be affected that then change what goes into the embryo and fetus, again affecting normal development. So we piggybacked our study on a birth cohort called TIDES. And TIDES was a study conducted at 4 sites in the U.S., California, Washington, Minnesota, and New York. They enrolled women in the first trimester of pregnancy and measured their phthalate exposures over the full pregnancy and also measured anogenital distance in the newborns, males and females. Anogenital distance is becoming a more popular measure in epidemiologic research on endocrine disruption, and it's basically the distance from the anus to the bottom of the genitalia in males and females. And there's 2 versions of it, a short and a long. This study, the main study, TIDES, was just published 2 weeks ago in Human Reproduction, lead author Shauna Swan. And they did find an association of the mom's urinary concentrations of phthalates and shorter anogenital distance in the male newborns. And this is confirmation of previous studies. And why we care about this measure, number one, in rodent studies, it's been well established that prenatal exposure does affect, prenatal exposure to phthalates can be measured in terms of shorter anogenital distance in the males at birth. And this shorter anogenital distance is predictive of the fertility of that male over his lifetime. In human studies, there's been correlations established between AGD in males and their sperm counts, testosterone levels, and also in adult females with their numbers of ovarian follicles. And then a recent study showed in the male mouse that in the postnatal male mouse, you can manipulate, using hormones and even castration, the distance of the AGD, the anus to the genitalia, suggesting that it has a plasticity to it and maybe could be altered. So the main data I'm going to show is this, which is we measured the association of the urinary phthalates and the AGD, or the HCG, the hormone. And of the 6 or 5 phthalates that we measured, the strongest correlation was with monobutyl phthalate. And the general trend was that with higher concentration of urinary phthalate, the mothers carrying the female fetuses had higher HCG relative to the mothers carrying male fetuses. And this was true for monobenzyl phthalate as well. And then when we did a comparison between the low exposed and the high exposed, the 10th and the 90th percentiles of urinary phthalates, we saw about a 15-fold difference between, again, the low and high urinary monobutyl phthalate levels. And then the next question we asked was, is the effect of phthalate on this placental hormone or pregnancy hormone playing a role in the ability of the phthalate to affect male development? So in a low to high comparison, again, of the mom's urinary phthalate levels, there was about 5-6% lower AGD in the males. And of this 5 to 6%, we estimate about 20 to 30% is due to the disruption in the HCG, or the placental hormone. So the implications of these findings, again, there needs to be more research, more understanding at the population level how these relationships work in the first trimester and how they relate to the outcomes in the babies at birth and beyond. But it offers a possibility to better utilize secreted placental hormones in the first trimester to gauge normal versus abnormal sex-specific fetal development, which could have implications for clinical practice, for population monitoring, for epidemiologic research. Secondly, in cases where we can identify abnormal hormonal profiles early in pregnancy, the question comes up, can we intervene sooner and possibly prevent long-term health effects in the child? And then lastly, I think our data is showing us that even though most of the literature and the research has been focused on male development, that phthalate exposure and placental hormones could be playing a role in female sex-specific development. So I'm going to end there with this image, and again, at the end, I'm happy to take questions.
Thank you. Okay, finally, we have a third-year grad student, well on her way to a successful career, I would say, give her presentation from Sherbrooke, Quebec.
Yeah, thank you. So thank you, and hello everyone. First, I wish to thank the Endocrine Society for giving me the opportunity to present some of the results from my PhD project. My results demonstrate that a perinatal exposure to a mixture of common phthalates and flame retardants lead to autistic feature in our rat model. I'll start my presentation with a few words about endocrine disruptors. So endocrine disruptors are chemicals which imitate natural hormones, such as thyroid hormones, like you see here. The structure are very similar. The two groups on which I focus my research are phthalates and PPDEs. So phthalates are plastic additives, so we find them in every plastic object like toys, packages, dishes, but we can also find them in more unexpected products like cosmetics or in the dust in our homes and schools. PPDs are flame retardants used to slow the emergence of flames when an object is about to catch fire. Therefore, we find them in objects at risk of catching fire, like cars, electronics, airplanes, but also a lot in households, furniture, mattresses, and textiles, including children's pajamas, for example. Infants and toddlers are the most exposed population, but also the most vulnerable because their brain is still in development, and that is why endocrine disruptors are a major preoccupation for children's health. When I began my project, several epidemiological studies had already linked exposure to several endocrine disruptors, including phthalates or PPDs, with behavioral changes related to pathologies like autism spectrum disorders, such as intellectual disabilities, immune dysfunction, anxiety, and with core symptoms of autism, especially social behavior. Unfortunately, epidemiological evidence can only point out an association between exposure to the compounds and effects. In order to demonstrate cause and effects, animal studies are necessary. Our study is the first to evaluate the combined effect of two very common groups of endocrine disruptors at doses relevant to a human exposure on the behavior of developing mammals. We use three different groups of animals. One group of pregnant females used as control, exposed to nothing. One group of pregnant females exposed once to valproic acid, an anti-epileptic drug which greatly increases the risk of autism in humans and is used as means of inducing autistic behavior in rats. And one group of pregnant females exposed during the end of the gestation and beginning of lactation by daily gavage to the mixture of 3 phthalates and 2 PPDs. After, we assess behavior of the rat pups in each group with behavioral tests relevant to detect ASD features. So, we look at maternal bonding, locomotor activity, explorative behavior, anxiety features, sensory motor gating, which is a key neurological process, and of course, socialization. Here is a brief summary of the results for each test with the comparison of the group exposed to the endocrine disruptor and our positive control for autistic behavior in rats. As you can see, the results are really concordant for the two groups in most tests, revealing less maternal bonding for males in both groups compared to control, hyperactivity for both groups, a high variability in sensory motor gating, which is a neurological process of filtering out redundant or unnecessary stimuli in the brain from all possible environmental stimuli. Several studies have characterized the same phenomenon in autistic patients. We also have an alteration of social behavior specific to males, and the only test results that are discordant for the two groups are the group exposed to endocrine disruptors show hyperactivity, while our valproic acid autism rat model showed anxiety. But both are symptoms present in autistic patients. As you can see, we observe behavioral changes in rats exposed to endocrine disruptors that are very similar to autistic features in humans. These findings are important because they identify preventable risk factors for one of the major public health concerns for which we have no prevention and no cure, autism. The ongoing increase in the prevalence of autism is really worrisome. In 2012, the CDC estimated that 1 child in 88 had an autism diagnosis in USA, and at the same time, Autism Speaks, a major advocacy association, estimated the cost of services for 2012 only for these children at $137 billion in the USA. Now, the latest estimation of the CDC for 2014 has now reached 1 autistic child in 64. In addition, because there is a strong sex ratio, 5 boys for 1 girl, It represents 1 autistic boy in 42 in the population. This exploding prevalence over the years, as well as the high sex ratio, point to an important role of the environment, and according to our findings, to endocrine disruptors. More and more studies point to the extreme sensitivity of the developing brain to endocrine disrupting chemicals found in our daily environment. Our findings show that we have to consider the combined effect of these numerous substances, even when present at low doses. Women of childbearing age should be aware of the risk of such exposition and consider ways to reduce their own exposure and the exposure of their future children. Because with some changes in our habits, we can reduce our exposure and then reduce the risk for the future children to develop autistic features. So, in conclusion, here are some examples of ways to reduce our daily exposure. Regularly clean and aerate the house, because these chemicals accumulate in dust, so you can also have an air purifying system. Avoid plastic utensils, and in particular, do not eat in microwave, for example, plastic containers with food in it, because these chemicals migrate from plastic into food, especially when the food is hot. And limit plastic exposition of the children by preferring wood toys instead of plastic toys. Avoid PVC floor. Do not put electronics like TV or computer in bedroom, especially in children's bedrooms. And finally, reduce the use of cosmetics, especially cosmetics for babies. These are a very simple example, but there is also a lot of others. And finally, I wish to thank every person involved in this project, my supervisors, and all the funding agency, and thank you everyone for your attention.
Thank you. Now we'll open this to questions. Also remember to use the microphone and also announce who you are and from what organization.
Hi, Jennifer. I'm from Frontline Medical News. How could, the last speaker spoke to this a little bit, but how could we intervene sooner to limit exposure in women who are pregnant? I mean, what can we do clinically that really matters?
Mm-hmm. Well, the data is suggesting, again, we need to collect more data and understand more at a population level how these secreted placental hormones do indeed vary by sex of the fetus, by maternal exposures, and by the outcomes in the child. But the idea is that if we can understand at a population level, can we establish, similar to as already has been established, say for HCG and Down syndrome, can we establish clinically meaningful cutoffs where they're likely to be abnormal versus normal development, and advise a woman appropriately? What would you advise them, though? That's the question. Well, I think the big question is the kinds of developmental effects that we're concerned about in measuring, how much plasticity are in those kinds of pathways and those types of processes. So the anogenital distance was recently studied in the postnatal period and reported to be plastic in terms of you can alter the hormonal environment of that male mouse and alter their anogenital distance. So the question is, in the fetal period, would you expect that same type of plasticity? And I mean, of course, talking about removing exposures is at a societal level, I think, is the first conversation, or just how much tolerance do we have as a society for these exposures. But assuming that we are seeing population trends, can we translate to that clinic in translate that knowledge to the clinic in terms of doing better prenatal screening, earlier than is currently now done, starts at 10 weeks. Can we do it earlier and identify, again, normal versus abnormal development?
Can we address it a slightly different way, maybe? I know Leo wants to.
That's not really where I'm thinking. I mean, that's a very dramatic way to think about it. I would think about ways to advise a woman to remove exposures, some of the low-hanging fruit in terms of where we know these chemicals are coming from. Again, I would prefer to see this addressed at a societal level, at a population level, but again, there's a lot we could study and learn from early periods in pregnancy that may be useful in advising women?
There are safe and simple steps that families can take to reduce exposure to the most harmful and prevalent EDCs of concern. They can eat organic, they can avoid microwaving plastic, they can simply open the windows in their homes to recirculate the air and prevent accumulation of the EDC dusts of concern that can accumulate from electronics, furniture, and other activities. And they can also avoid canned food consumption, which is well documented to lead to increases in bisphenol A. So one neither needs a PhD in chemistry, nor needs to consider hormonal correction for the health effects associated with EDCs. As a pediatrician, as someone who works closely with obstetricians, I think that would be a dangerous response to go into a compensatory mode and use a drug to treat an exposure that can simply be eradicated through personal behavior change and regulatory change.
I'd like to add actually one very short comment to follow up on that. I think the other thing that has to happen is that there have to be better ways for physicians and patients to talk to each other. So pregnant women speaking to their OBGYN, they ask them the right questions. What's your lifestyle like? When they find out what they eat, make the recommendation, try to eat organic vegetables, wash your vegetables to get the pesticides out. So I think that physicians can play a real role in helping people make some very small lifestyle changes that could probably make some big differences.
Hi, I'm Ed Sussman with MedPage Today. Listening to this, I think, wow, how did I manage to live this long? And have normal children. And
you think?
And you all would be the age of my children. So I'm looking for the collapse of humanity, and I don't see it worldwide. Convince me that this is really a problem.
Go ahead.
So we spoke to that in our report, and this also speaks to the report by the World Health Organization and the United Nations Environment Program that documented the urgent problem here. We've documented across Europe and across the world, in some cases, rates of fertility are dropping below the replacement threshold, such that we will have subsequent population decreases. And you are right. In many cases, this is a silent epidemic, but it's a real epidemic supported by a substantial mountain of human and laboratory evidence suggesting that endocrine-disrupting chemicals disrupt a broad array of hormonal processes with implications for human health.
So I think also, if I can, I'll just chime in here very quickly. But one of the things that I hear often is this concept that if there isn't overt toxicity, then you don't really have a problem. And I think one of the things we're seeing with this whole issue of endocrine disruption is we see effects on the population level that are not consistent with overt toxicity, like death or, in some cases, cancer. Think about, for example, a decrease in IQ levels. That's not something that is visible kind of on the surface of the street. You are taking away human potential, though, and this is very well documented that PCBs and other chemicals have this effect. I think that, we need to begin to think a little bit in a more sophisticated way than this concept that there's either abnormality or normality that is visible on the face of your children, because that's not what's happening. Andrea.
Yeah, I was going to make a similar comment. When you think about the distribution of any disorder, you have some sort of curve, and I think what's happening with the endocrine disruptors is they're pushing that curve a little bit farther to the side that means you're more likely, maybe just a little more likely, to get a disease such as type 2 diabetes, or to be obese, or to be infertile. So as an individual, maybe it won't matter for me, but when you think about the 7 billion people on the planet, that's a lot of people where a 5% shift is adding to the disease burden, the cost, but also the individual burden of those people who actually have these problems, but on the surface seem fine.
Dr. Trisandi, you mentioned the low rates of fertility in certain countries, but isn't that due to social change? In fact, when you become wealthier, you tend to have fewer children, and even when in societies where you don't have a lot of resources, you tend to have fewer children. Are these more social changes rather than chemical changes?
No one would disagree with the notion that there are social changes that have occurred over the past 30 years and that families make decisions, but there's a substantial increase subsequently in the need for assisted reproductive technology for families who try to conceive a pregnancy but cannot seem to do so. And in our report, we attribute a substantial burden of male infertility contributing to the need for assisted reproductive technology, with the resultant cost in the range of 4.7 billion euro annually.
I just don't know that you have a cause and effect here.
There's substantial evidence. Clearly, these conditions, as I described in my presentation, are multifactorial. And clearly, I think we're talking about two different pictures. In the broader picture, there are clearly ecological trends in behavior that contribute to decisions about fertility and about reproduction. But there are many families who are trying to have a pregnancy, have a healthy child, who are having increased difficulties on the margin due to uncontrolled exposure to endocrine-disrupting chemicals in the environment.
One from the web, and I'll come to you right after. I think this one is still from the Brussels group, and this is a question for you, Dr. Trisande. It says, France is the third biggest user of pesticides in the world. Should it be one of our major source of concerns regarding brain development?
It's fair to say that organophosphate and other pesticides contributed the largest percentage of the economic costs we're able to attribute to EDCs in Europe. Insofar as France is a disproportionate contributor to pesticide exposure across Europe, France may have a disproportionate share of the economic cost of EDC exposure. And those costs, human and health costs, need to be compared side by side with decisions about safer alternative approaches to farming, eradication of unwanted pests in homes, and other uses for which historically pesticides have been placed.
I'm not convinced on the reproductive, because I think people just, now that we have the technology, people try it. And so it would seem to me that the need for more reproductive IVT, for example, is because it's there. And people just didn't have children before. And so it's, I mean, since we've had IVT, I mean, have you tracked it within that? I mean, and even that's advanced so much that I'm not sure you can draw that conclusion.
I'm not suggesting that 100% of ART is due to endocrine disruption. Quite the contrary. It's a modest sub-percentage of all of these chronic conditions that are attributed to EDCs. For example, with childhood obesity, the attributable portion of BPA was on the order of 1.5%. Similarly, for EDCs, we attributed a small percentage of ART. I can't give you the number offhand because I don't have all the statistics in my brain. My brain can only have so much space. But suffice it to say that we are not arguing for even 30 or 50% of these diseases to be directly linked to EDCs. But if you look more generally at the various reports of the World Health Organization and the United Nations Environment Program, they support the notion that a substantial contribution to human disease today is due to environment. And within that, a substantial subproportion is endocrine disrupting chemicals.
So I've been told that our time is pretty much out. This is just the beginning of a long conversation that the Endocrine Society and these members in particular have been engaged in. I really appreciate you coming, and thank you for your participation.